I guess I'll begin. Anybody in here who's not undergoing cell division right now?
Undergoing a what?
Who's not undergoing cell division? Is there anybody in here?
No.
Good, you beat that. Okay. We're multicellular organisms. Well, even if we were single-celled, we would probably be making copies of ourselves through asexual reproduction. But we have a lot of cells, and most of them are up to no good. But our cells need to undergo lots of cell division. First of all, following reproduction and fertilization, you have a zygote. It's a single cell. The diploid state has been restored, so you have homologous chromosomes, one from mom, one from dad. And then from that, the single cell develops into an embryo and eventually into an infant who gets born. Now, during that whole time, obviously, there's cell division going on. And it's highly choreographed. In other words, you just can't, one cell, as a different, let's say, remember, we're differentiating into all different kinds of cells. We've got fat cells. We've got muscle cells. We've got red blood cells. We've got white blood cells. We've got neurons. We've got pancreatic cells, all different kinds of cells. All those have to differentiate into different types during development. Plus, there's this whole thing about a body plan. So we're tetrapods, right? There's genes controlling that. At the same time, all these different systems are being put in place. So every time you have cell division, those cells need oxygen and nutrients. There's growth factors that initiate blood vessel formation, bone formation, all that kind of stuff. And that's all well and good. But it's highly choreographed and constrained. You can't have a cell that decides, I'm lonely and I have a mutation and I want a whole bunch of friends of mine that are just like me, and start reproducing wildly, as what happens with cancer. So, in fact, cancer is what you would actually expect in a multicellular organism, unless there was some way to stop it. Okay, so we're first going to start with cell division. As you know, we have chromosomes. These are homologous chromosomes. That means we have one from mom and one from dad. Say this one is from dad, and this one is from mom. And when there's actually cell division, you need to double up the number of copies. Okay? So we end up with DNA replication. And we can see that we have big A, little a, little a. Most of the time, our DNA in our nucleus, you can't see it. It's not condensed. It's widely spread out. It's only during replication that DNA actually condenses into the chromosomes that we can actually see. Now, when there's DNA replication, it means that you are producing identical daughter cells. Those identical daughter cells are diploid. That means the one original cell has homologous chromosomes, one from mom, one from dad. Same thing with the daughter cell that's produced. This has nothing to do with reproduction. This has to do with cell replication. Don't get this confused with meiosis and reproduction. Okay, so this is uncondensed DNA in the nucleus. It doesn't look like okay, it doesn't matter. We've got this going on. Okay, so now the thing is, when we're actually replicating, it'd be difficult to divvy up the DNA if we replicated all this in this form where it's all spread out. So what happens is, we need to replicate the DNA, and the first start of the process is condensing the DNA. It's easier when it's condensed into actual chromosomes, which we can actually see. And then, during cell replication, at one point, we divide the copied DNA. So when we make a copy of the DNA during cell replication, we basically end up with two copies of the complete genome. And one goes to one cell and one goes to the other. And then that's what you have in terms of daughter cells. They're clones of each other. They're exactly the same. So I have skin cells. Actually skin cells. And when they start dividing, and they're dividing a lot right now, the skin epithelial cells are, they don't have a very long life, so they have to be renewed all the time. The daughter cells are identical. Remember, every cell has the complete genome. Not just the cell, not just the genes for the cell type that it is. And this is what chromosomes look like. This is what a diploid chromosome looks like. They're homologous chromosomes. Why are they that way? Who do we get these chromosomes from? Anybody? Mom, Dad. Right. So, we'll say this is Mom, and we'll say this is Dad. Okay? Now, the whole chromosome is this whole, that's not going to work, is going to be this whole thing. Each separate section is called a chromatid, And you end up with 2 chromatids per chromosome. One from mom, one from dad. All this gets replicated. Oh, they're also connected right here. It's called the centromere. And all this has to get replicated during cell division. So it gets condensed. Okay. Okay. Now, as I mentioned, cell division and replication is highly choreographed. You can't have cells going off on their own and start replicating. It's a no-no. So there's all these systems in place to stop that from happening. This is a cell cycle. Most cells spend 99% of their life in the G1 phase. Cell growth and function. So they need a certain amount of nutrients and all that, and they function. They do what different cells do. Muscle cells have contractile powers. Pancreatic cells make insulin. Neurons carry electrical signals. Genes are expressed or not expressed. Not exactly on and off. It's more like making more protein versus less protein, all that kind of stuff. So this whole thing, by the way, is called interphase. That means it's G1 phase, S phase, and then G2 phase. Mitosis is not part of interphase. As I said, most cells spend all their time in G1 phase. But if a cell gets a signal to replicate, then since each cell has a full complement of all the chromosomes, we have 2 cells, we need to replicate all the DNA. And that's called S phase, synthesis, DNA synthesis. And so all the DNA, before it's turned into, condenses into chromosomes, all that DNA is replicated in the S phase. And then in G2, cell needs, not just do we need the DNA replicated, but we need everything in the cell replicated. Mitochondria, all this other machinery that's in it, plus enough resources to do that. So that's what's happening with G2. You're growing these cells bigger and bigger, more resources, and preparing for entry into mitosis. Okay, so S phase, G1, S, G2, and then mitosis. Okay, so some cells, most of the time they're G1, but sometimes they receive a signal, for whatever reason, to divide. So here it is again. We have interphase, and then this whole thing is mitosis. The first phase is called prophase. The second is called metaphase, where all the chromosomes, remember there's 2 copies of everything now, line up along the center of the cell. They're connected by these microtubules. And then during anaphase, they're pulled apart. And then finally, during teleophase, the nuclear envelope reforms and the cell finally divides. And that's mitosis. And this phase is also called, that's cytokinesis. Okay, and here we are again. So during prophase, the chromatin, that's the condensed DNA. The nucleolus starts to disappear. The mitotic spindle fibers start to form. Prometaphase, you can see the chromosomes clearly. Sister chromatids, and then metaphase along the central line. You can actually see these microtubules. Okay, they come up from both sides. And then during anaphase, the sister chromatids and chromosomes are pulled apart. And then finally, teleophase and cytokinesis, you end up with 2 identical cells. Okay, same thing with animals. Okay. Why do you think cells divide? One reason. Any reason here? Huh? I'm dead.
Getting injured?
Injured. Yep. Okay. Anything else? Do you think cells last forever? No. What happens? When you drive a car, and finally you end up with 150,000 miles, things start to break and wear and tear. Same thing happens to cells. Okay. And then finally, actually, initially, once we have cellular reproduction, sexual reproduction, we've got a zygote, which is a fertilized egg from a sperm and an egg. Well, that eventually turns into a multicellular organism of some sort, if it's that kind of thing. So, an animal, a plant, fungi, something like that. And then it's got to go through this whole developmental process. And so those are the kind of the 3 main reasons that cells undergo division. Now, furthermore, cells have different lifespans. They wear out, they get injured, but some cells are constantly being renewed. Our epithelial skin cells are undergoing division right now, on and on. You can't feel it, but it's happening. They don't last, they're not alive for very long, only a couple of days, and then they're replaced. Red blood cells, they're actually being made all the time. They have to go through maturity, so you start with stem cells, they get turned into red blood cells, and then those are being replaced as well. And then a lot of cells like muscle cells or other organs, they don't get replaced very often. They do their function, they remain intact. Some cells almost never get replaced, like neurons. There is some, but not much. Now, some cells are bad cells. They might get lonely or something like that, and they want individuals, well, all the cells are just, remember, they all share identical DNA, but, you know, when that DNA is copied, sometimes there's mistakes and there's mutations. And when those mutations, not just one, but usually a number of different mutations happen, they go rogue. In other words, they start to uncontrollably replicate. And that's what cancer is. Cancer, the mutations happen in mature cells, not in gametes. Okay? So a mutation happens, let's say it develops tumors, that's not going to be passed on from one person to the next. It's just going to be passed on to daughter cells. So cancer, I guess you could say, is one of the predicted outcomes that would happen with multicellular organisms. The same thing would actually happen with single cell. Cells need a certain amount of nutrients and all that stuff to reproduce, and you could see a mutation in a single cell organism that makes it replicate when it's really not ready to. It wouldn't be good, it would die. But in a multicellular organism like our cells, we've got all this cell division going on all the time. Occasionally, there's going to be some mistakes, mutations. There's a bunch of different things that might cause these mutations. We'll get to that in a little bit. And if things go wrong, then we're going to end up with uncontrolled cellular replication. There are essentially what is happening is cells inside the body of a multicellular organism have given up their own sovereignty. They can't just say, oh, they're not saying this, actually, they can't think, but they're not saying, oh, I want to replicate. In fact, they forego replication unless they get a specific signal. If they don't do that, there's kind of like a police force that comes along and stops them from doing that. Now, as I mentioned, during development, we have all this cell differentiation into different types of cells. It starts out with stem cells that then give rise to other kinds of cells. So, Here's a zygote. Here's what's called the blastocyst stage, and then here we have an infant multicellular still undergoing development and differentiation. Okay. There are certain genes. Remember we talked about gene expression. It's not exactly like an on-off switch. It's more like how much protein is being made. So it's like a dimmer switch, right? It's not doing it. But anyway, you get the idea. So there are some genes that there might be a mistake, and let's say they get turned on, or they're starting to express some protein and they shouldn't, and they keep replicating. Well, before that happens, part of the whole cell cycle and replication are these genes that normally get turned on when they get a certain signal. And they're called proto-oncogenes. Now, onco means cancer, malignancy. Proto means they're not cancerous. In other words, their function is not to turn into cancer. Their function is for replication. I think it's actually a bad word because it implies that these genes, like, that's their function. They're not. Normally, these proto-oncogenes only get turned on for replication, otherwise they're off. They need a signal, whether from a hormone or some other protein, to start dividing. Now, if the cell replication process isn't stopped at appropriate time, or it starts and it shouldn't, basically you have cells that have broken out of their kind of self-imposed control, and it's kind of like piracy. Usually what happens is, even when this happens, it stops from happening. But occasionally, it's not. We talked about evolution, we talked about how individuals are affected by selection, the selective environment, let's say, and then populations evolve. Well, you can actually do the same thing with cells. Different scales. So instead of treating the individual of a multicellular cellular organism as an individual, you instead treat each of the individual cells as individuals. Cells that start reproducing uncontrollably, compared to all the other cells, have higher fitness. Now you're thinking, fitness. Fitness, that means passing your genes on to the next generation. That's what those cells are doing. Even if ultimately that uncontrolled replication turns into full-blown cancer and kills the individual and those cells. So it's the same thing in terms of fitness. A mutation that lets a cell replicate uncontrollably increases that cell's fitness in regards to all the other cells around it. In the end, it's going to kill that cell and all the other cells around it, but it doesn't know that. It has a mutation, it starts dividing uncontrollably, it leaves more daughter copies than all the other cells in the body, takes up all the other resources. You end up with tumors, eventually it kills the person that has them. Now, we normally think of fitness as something that's a good thing that increases an individual's survival and reproduction. Well, if you look at it from the cellular level, it's doing that, even if it ultimately ends up killing the individual and all the cells in it. Okay. So, as I mentioned, natural selection, evolution by natural selection, can work on individual cells. In other words, cells that can replicate, even when they're not supposed to. That's natural selection, evolution by natural selection, even if it's eventually going to kill them. And those populations of cells evolve. In fact, they evolve and mutate. Usually cancer doesn't start with just one mutation. It starts with one and then another accumulates and another until finally you have enough mutations that you end up with cancer. Okay. Now, unless the cell gets a signal to replicate, it doesn't. It needs a specific signal, usually some sort of protein, that initiates cell replication for whatever the reasons that we normally think about, wear and tear and injury and all that kind of stuff. Furthermore, we have a police force. So, think about a city, our city. Now, most of us don't start running around bashing people in the head, right? We're not like that. We're nice people. Some of us are. Like me, not so much, but, you know, it takes all kinds. Okay, so normally I'm well behaved. I don't go crazy. Though I do have a problem. I don't really have very good morals. So on the weekend I go out and I start raping and pillaging. Not a nice thing to do. Now, because I'm not controlling myself, there are people out there called police, and if they catch me raping and pillaging, they're going to beat the crap out of me. Then they're going to hold me into jail. Then they're going to put me in jail. It's going to be a mess. But you'll be protected from me. Well, the body has the same sort of police force. Now, as I mentioned, most mutations happen during your lifetime in somatic cells or autosomes. These are all our cells that have nothing to do with sexual reproduction. The cells that have to do with sexual reproduction are called our germline cells. So, because there's all this replication going on all the time, occasionally you end up in mutation and it generates tumors and all that kind of stuff. Okay, so most of those mutations occur in your somatic or autosomal cells. Only some mutations are actually inherited. In other words, you can inherit cells or genes that mostly lead to cancer. Inheritance of cancer-causing genes from parents to offspring kind of gives those cells that have that inherited mutation a head start. Anybody know what BRCA1 is? Okay. It's the gene, it's a tumor suppressor gene, and it's responsible, if it's mutated, when passed from parent to offspring, it increases a woman's risk of getting breast cancer, ovarian cancer, and also prostate cancer for men, among others. It's part of the police force. So BRCA is a tumor suppressor gene. It's part of the police force. It creates a protein, BRCA, and when it detects a cell that's not behaving properly, it comes along and it shoots it. Actually, it doesn't do that. It hands a gun to the cell and says, you have to kill yourself. And that's what the cell does. It commits suicide. Not exactly just because it's such a nice guy, but just because that's what's going to happen. Okay. So, cancer is essentially a genetic disease because we're dealing with DNA here, and we're dealing with mutations in the DNA. Now, we usually make a distinction between genetic in terms of inheritance and genetic in terms of mutations. Well, the only inherited genetics are in our germline cells, like BRCA. So, if someone inherits a mutated copy, which is a tumor suppressor gene, and it doesn't stop other cells from replicating uncontrollably, they have a higher incidence of cancer. In other words, they're faced with oftentimes a terrible choice. So we can say genetics in terms of inheritance, or we can say at the molecular level, genetics in terms of mutations that happen during replication that will lead to cancer. Cancer is almost essentially environmental, even when it's inherited. The cellular environment and the genetic environment. But for the most part, environmental, there are things that cause cells to mutate and to become cancerous. Solar radiation. Certain chemicals like benzene. Cigarette smoking, a whole slew of different chemicals. Viruses. Some viruses can start cancer going too. Is contagious. At the moment, there's nothing, no kinds of cancer for humans that is contagious. In other words, you grab someone, you start smooching, and you pass it on. No, it doesn't work quite like that. You can pass on a virus that causes cancer, but you can't actually transfer cancer cells from one person to the next. But there are some organisms where that does happen. It seems to be fairly common in clams for some reason. It also, dogs get a kind of non-serious, very slow-growing kind of cancer that they can transfer to each other. In other words, they transfer cells, their skin cells, from one to another, and it starts cancer on the other individual. There's a carnivorous marsupial in Tasmania, off the coast of Australia, called Tasmanian devil. They have wild, violent sex, where they tend to bite each other a lot. Well, a kind of cancer where they can control, cells from one cancer on an individual can be transferred to another. That started a number of years ago, and it's almost turned the whole population extinct because it's a very aggressive cancer. They've saved enough to put them on separate islands so that they're not infected. So there might be, and furthermore, that's a pretty strong selective force, as you can imagine. There appear to be some individuals that have inherited a mutation that made them less susceptible to this kind of cancer. So it might not be the end. Is cancer on the rise? Depends on the kind of cancer. Most common kinds of cancer, no. But certainly we have a lot more synthetic chemicals in the environment that we put in there that are cancer causing. They're carcinogens. The immune system, that's what it's there. That's part of what it's there for. It's there to protect you from cancer as well as other parts of the immune system to protect you from infection by infectious diseases like bacteria and viruses. Is the treatment worse than the disease? Sometimes. In fact, basically, for the normal cancer chemotherapy treatment or radiation was that those things would kill cancer cells hopefully faster than it killed your own cells. But sometimes the dosage was wrong, and so a lot of people ended up dying from the treatment. Okay, so cell cycle control. Remember, we're talking about mutations here. Forget about, in the germline, just regular mutations in skin cells, in pancreatic cells, in kidney cells, all that. Now, remember, gene expression is usually not a matter of being turned on or off, but it can be. You can actually, in fact, we talked about proto-oncogenes and oncogenes. Well, those genes are the genes that are turned on, they're activated. When they should probably get a signal at a certain point to stop producing whatever proteins they're producing. So, genes that encode proteins that promote cell division are called proto. Okay. So which kind of mutation will cause the most trouble? A mutation that activates a inactivates a proto-oncogene or activates? Okay. If a cell is reproducing uncontrollably, these proteins see it and they stop the cell in its tracks. What they actually do is they make the cell commit suicide. So what do you think, activate or inactivate? Yeah, good. Okay. Okay. And cell suicide is called apoptosis. Okay. So the protein, usually on some immune cell, tells the cell, you have to kill yourself. And it does. And so the cell starts basically coming apart at the seams. Okay. So with the BRCA1 protein, tumor suppressor gene, if it detects a cell that's acting inappropriately, it makes that cell no, I don't have it. Hold on. I'm dying. Okay. I think it's my tumor suppressor genes are telling me to die. Stop it. Okay. Now, since it's a tumor suppressor gene, if there's actually a mutation in BRCA, what is it not doing? It's not telling cells that start acting badly to die. That means it allows those cells, if there's a mutation, to keep living. And it allows that cell, as it keeps replicating, to develop more and more mutations and accumulate more mutations. And that's usually what starts with cancer. It's usually not one mutation, it's usually many. Okay, so a tumor, God, I hate them. But anyway, is a mass of cells that are replicating without being stopped. It doesn't mean that it's cancerous tumor. It can be a benign tumor. Now, we differentiate because normally when a cell comes up against another cell, it recognizes that other cell, and there's something called contact inhibition. And that stops the cell from replicating and growing. So if you get a tumor developing in one place, but it doesn't keep growing as it hits other cells, it's called a benign tumor. Contact inhibition has stopped it from continuing to replicate. It doesn't mean that a benign tumor can't be dangerous. It can. There are certain pituitary tumors in the brain that are very difficult to operate on and can be fatal, but it's not cancer. It's only when cells lose contact inhibition, they grow next to another cell, and instead of stopping, they actually start growing into the tissue of these adjacent cells. So they've lost contact inhibition, and now they're actually cancerous. It doesn't mean necessarily that they're all that aggressive. They can be growing slowly, and you could probably live with it for the rest of your life without suffering too badly. So some prostate tumors are like that, others are not. So a tumor can start. It cannot be invasive. In other words, it maintains contact inhibition. It's a benign tumor. If it's invasive, then it's malignant. Now, furthermore, cancer can be invasive, but it can still only grow into, let's say, surrounding tissue, not move. But some cancer can. called metastatic cancer. So you get lung cancers are pretty metastatic in general. You end up getting them primarily in the lung, but then you can get them in the bone, in the brain. These are all secondary cancers because they've moved. What they've done is they've lost something called anchorage dependence. So when our cells, normally they're part of a tissue, they're all hung together, they don't move at all, right? But in this case, a cell might decide it's time to go on a vacation to the brain. I want to see what it's thinking. So it hops a ride in the bloodstream or the lymphatic system and starts wandering around in the brain and then decides, I'm lonely, I want a bunch of cells just like me with my mutations. It starts replicating, and now we've got a metastatic brain cancer. Okay? So then we're down to this. Non-metastatic, it doesn't travel. Metastatic, it does. So this is the basic progression. We start with normal cells. As they divide, they get instructions to divide. They get instructions to stop dividing, all that. If there's some mutations and there's unregulated cell division, some divide, but they're benign. They have contact inhibition, they don't invade surrounding tissue. Some do, and these are malignant. And then some actually not only invade surrounding tissue, but can travel throughout the body, creating secondary tumors. Okay. So a carcinogen is any substance that can cause mutations that lead to cancer. There are also going to be things called mutagens that just cause mutations that then subsequently lead to cancer. There's all kinds of carcinogens all over the place. Some of them are completely natural, like in cigarette smoke. There are some fungi that secrete chemicals that are carcinogens. There are a lot of man-made chemicals that are carcinogens, like benzene, parts of gasoline and things like that. There are a lot of chemicals that plants secrete that are also carcinogens. They use them in their seeds to protect the seeds from being eaten by something else. So a carcinogen is any substance that does something. Now, we've heard about free radicals and kind of ions that are highly reactive. And one of the ways that cigarette smoke and the chemicals in it cause cancer is because of free radicals. But they've actually recently gone through and shown exactly the kinds of mutations that eventually lead to lung cancer. Most lung cancer is due to smoking, but not all. Also, a bunch of different other kinds of cancer, including oral cancer, throat cancer, and esophageal cancer. Colon cancer, a bunch of others as well. Carcinogens, so if you're exposed to carcinogens, you're increasing your risk of initiating a cancerous tumor. UV light, so skin cancers. Ionizing radiation, from radioactive substances, a whole bunch of other things. Some diets protect you against cancer and some don't. And then there's always also some cancers that are genetically inherited. Lack of exercise can increase your risk of cancer, but also you get older, most cancers happen in older people. It's like your immune system, tumor suppressor genes, they start falling down on the job, not doing what they're supposed to do. Okay, this is where we left off. How do tumor suppressor genes actually stop cancer cells from dividing? Guys, remember? There's a word that goes with it. It starts with an A. I heard it. Apoptosis. Okay. So what happens is, if a cell is damaged, something is wrong, there's usually proteins that are on its cell surface indicating something is wrong. There are these molecules that, well, there's parts of the immune system like lymphocytes, T4 cells and things like that, and macrophages that come along, and when they detect this damage, they release a chemical that binds to these aberrant proteins, and then it makes the cell decide to kill itself. And that's exactly what it does. So that's primarily what tumor suppressor genes do. When a cell is not right, it says it's time for you to go. Now, and that's apoptosis. What is it that, what do we call it when we say that a cell can actually, why can a cell invade surrounding tissue? What is there a loss of that allows cells to keep growing into other tissue? Anybody remember? Starts with a C. Remember, huh? Okay. It's called contact inhibition. Okay. What is there a loss of when a cell starts moving around the body? Remember? Yeah. Thank you. Okay. Okay. Often we see cancer patients with, they lose weight. They lose lots of weight. And the mechanism behind that involved, now remember, cancer cells, remember, they're part of the body. They're normal cells, except that they're unregulated in terms of cell replication. Now when you have an embryo and it starts dividing, cells need nutrients and oxygen to do their functions. And normally in our body, the way it happens is that we've got all these circulatory, we've got blood vessels and arteries and all that stuff to bring these things to cells. Glucose, so they can produce ATP, do this work, all that kind of stuff. Now, creating blood vessels, so let's, for example, say you cut yourself. There will be an automatic response where those blood vessels will repair themselves, or you might actually make new blood vessels. And that's called angiogenesis. Similarly, patients have heart attacks. The body starts producing new blood vessels to kind of bypass that area. Cancer cells have the same ability. They release, like all these cells do that are damaged or whatever, they release growth factors when cells are not getting enough nutrients and oxygen, and that induces angiogenesis, or the growing of new blood vessels. Cancer does this too. Now, remember, cancer is replicating all the time, uncontrollably. The cell cycle that goes through it, around and around and around, that requires a lot of energy, a lot of oxygen. The only way to get that is that cancer secretes these growth factors and creates its own blood vessel supply and blood supply. And so essentially now most cells are not undergoing replication all the time, so they don't need all that new blood supply, but cancer does. The bigger the tumor, the more the blood supply it needs. And so cancer starts getting this blood supply to grow in the form of blood vessels. And in fact, in terms of chemotherapy, which is drugs that are used to kill cancer cells, some chemotherapy drugs actually stop angiogenesis. In other words, to starve the tumor. Remember, cancer cells are undergoing the cell cycle constantly, whereas most other cells are not. And so something like that is mainly going to stop the growth of new blood vessels. Our other cells don't really need it that much. They've already got their blood supply. So that's one way to actually stop cancer, is to starve the tumor by stopping angiogenesis. Now, if you remember, cancer cells, they're part of our body. We might have a malignancy from pancreatic cells, right? They're growing. our normal pancreatic cells, we have this immune system that is always searching for cells that are doing some bad things. Now, occasionally our immune system starts attacking the body itself, and that's the result. The results are autoimmune diseases like multiple sclerosis, like rheumatoid arthritis, like Crohn's disease. What the body is doing is it's recognizing these as foreign instead of part of your own tissue. And it starts attacking it, releasing antibodies. So the whole immune response. Type 1 diabetes is another example, right? Where it kills the insulin-producing beta cells in the pancreas. Well, our normal cells stop this process from happening by a whole bunch of different mechanisms. They secrete immunosuppressive molecules so that, let's say, a macrophage comes along searching for a cell that's doing bad things. These molecules downregulate it and make it not recognize something as non-self. These immunosuppressor cells are, let's see, what is it? Oh, anti-tumor T cells, dendritic cells, a bunch of different things. So one of the reasons, of course, normally what happens is with cancer cells, new cancer cells, the body, in fact, our immune system does recognize these cells as malignant, as non-self. And the immune system goes and attacks it with a tumor suppressor system. And it makes these cells undergo apoptosis. But because cancer cells were originally our own cells, they can suppress this from actually happening. So cancer cells have their own immunosuppressor chemicals and that stop other cells from actually making it undergo apoptosis. That's why it's often so difficult to actually treat. You would think, all we have to do is upregulate the immune system for cancer, and that should stop the cancer. But because of these immunosuppressive properties, it often doesn't work. Now, researchers at this point are learning how to do that, to get these systems actually operating and killing cancer cells. We'll get to that later in terms of treatment. Okay. Now, I mentioned this before. When we talk about evolution, we say selection operates on the individual. Individual has certain traits. Some are more likely to help it survive or reproduce than others. So, a cheetah chasing a gazelle. Some gazelles can escape the cheetah because they're faster. They can change direction faster. Whereas others can't. The selective force is the cheetah. Same thing happens reverse onto the cheetah by the how fast the gazelle is. Well, instead of individual organisms, we can look at tumor cells as individual cells. And with cancer, what happens is some cells get a mutation or a number of mutations that allow them to keep replicating. In other words, creating more and more cancer cells. In other words, they're leaving more offspring than normal cells, which, because of the cell cycle control, stop that from actually happening. So cancer cells have a mutation, that enhances their individual fitness compared to all the other cells that are behaving themselves. I realize that it seems like, well, how can that fitness is supposed to increase survival and reproduction? Well, that sort of thing normally eventually kills the organism with the cancer cells and eventually kills the cancer cells. Doesn't make sense, but evolution doesn't work that way. Cells have certain characteristics, and it allows them to replicate rapidly and produce lots of tumor cells. It's going to keep on doing it. So, much of cancer progression, it doesn't start with, it's not just one mutation. One mutation usually don't end up with a cell that's cancerous. Usually it takes a number of different mutations over time before a cell finally becomes malignant and does all the things that malignant cells do. In other words, cancer cells, because they're replicating all the time, they're generating new mutations. Most mutations might have no effect on the cancer cell, or they might be bad, but some might help it, let's say, metastasize, where the cells from which it previously replicated didn't have that ability. So, cancer starts, what we have is a model or a hypothesis that it's called multiple hit model. That means it takes many mutations to actually cause cancer and not just a single mutation. Okay. Cancer detection. Well, some cancers are fairly easy to detect. Multiple melanoma. It's a skin cancer. It's a very aggressive cancer. If you catch it before it gets beyond, let's say, stage 2, usually the person is going to survive. It's easy to get rid of. But once it gets into stage 3 or 4, it's an aggressive metastatic cancer that starts wandering all over the place and creating tumors in the brain and the bones, all sorts of different things. And so, let's say you develop a melanoma on your scalp. You don't check it, no one sees it. It grows and grows and grows. And then it's finally detected. Maybe it's generated tumors elsewhere. By that time, usually within a year, the person is actually going to die from melanoma. Most melanoma is caused by solar radiation. And so you can protect yourself, of course, by using sunscreen and all that. There's also a lot of other skin cancers that are much less aggressive. In fact, they won't kill you at all. So, skin cancers are fairly detectable. But there's other cancers that aren't, like pancreatic cancer is a great example. Usually by the time you start having problems, it's already invaded adjacent tissue, like the liver, so it starts in the pancreas, the liver, the intestines, all kinds of different places. And by then it's too late. So the secret to actually cancer is cancer prevention rather than cancer treatment. If you can detect it before it becomes serious, your chances of survival are much higher. Now, so let's say you have a lesion looks like maybe it's a little bit of skin cancer. How do you find out? Well, you cut a little piece of it off, the biopsy, and you look at it under the microscope. And you can actually tell whether it's a cancerous cell, because cancers look a little different than normal cells. And you can say, oh, this is melanoma. You can then treat it. You can also do biopsies. Let's say you do a blood test, standard blood test once a year, and your liver panel enzymes come back and they're elevated. Say, what's going on here? Well, you can actually go in with a nice long needle and plunge it into your liver and take liver biopsy. You can do that with prostate gland as well. So, furthermore, some cancers release certain chemicals or cause certain chemicals to release into the bloodstream, indicating that there's a problem. Like, for example, when patients have a heart attack, they release a protein that's the product of cell death from the heart muscles. And so you can actually diagnose a heart attack that way. With prostate cancer, there's something called PSA. It's a protein. Now, PSA levels can also spike because as men get older, their prostates tend to grow, and they start having prostate problems. And it's not cancer; it's just getting a larger prostate, and so the PSA levels might go up and might not be the result of cancer. But it might be, and so if you check for that, and most men, they get their PSA levels checked yearly when they get to a certain age. Their PSA levels have increased. They oftentimes will go in and do a biopsy of the prostate gland. Now, I will say that there are a lot of prostate tumors that are not very aggressive. In fact, there are some that are so slow growing that it's not worth taking them out and removing them surgically or using radiation or anything like that. It'll happen, let's say, in a man who's 70, let's say. Well, he's likely to die of something else, like a heart attack or a stroke, instead of that prostate tumor in 15 years, because it won't really grow all that much. Some prostate cancers are more aggressive. So, you obviously don't really want to treat a tumor that's not going to kill a patient, because prostate surgery, there's all these terrible things can happen with it. You can be incontinent. You can have loss of the ability to get an erection. All these terrible things. So you really don't want to go through prostate removal when you have a small cancer that isn't going anywhere. So there's this big debate about when you detect something, is it really good to treat it or just monitor it? Okay. Now, so Steve Jobs. Steve Jobs doesn't look like a hippie, but he is. And he was. When he was younger, he and Steve Wozniak, when they were starting Apple, they both had long hair. They took lots of LSD. They had some great ideas. But because of this, he had some ideas about health. And one of these ideas was that he was terrified of cancer and then removing it through surgery and chemotherapy. So he got pancreatic cancer. Now normally pancreatic cancer is a very aggressive cancer, but he had a kind that's much more rare, but also much more treatable. It's actually much more slow growing. So instead of going and having surgery and chemotherapy, he decided to treat it with herbal medicine and things like that. It was a big mistake. Eventually he had surgery and chemotherapy. Made him really sick. It kept the cancer at bay for a long time, like 7 years. But eventually, because he had waited so long, it killed him. Which is unfortunate. So if he had gone in, they detected it, it's a slow-growing pancreatic cancer, they had treated it properly, he probably would still be with us today. Okay. Now, you've probably heard of radiation therapy for cancer. It's better than it used to be. In the past, let's say 30 or 40 years ago, the radiation beams were wide. They killed a lot of adjacent tissue, made people very sick, and is terribly disfiguring. But nowadays, what they do is they can concentrate high X-ray beams strictly on the cancer and damage the cancer cells. So the radiation itself doesn't necessarily kill the cancer cells. What it does is it damages them, and then the tumor suppressor system that we've learned about recognizes damaged cells, and then those tumor suppressor proteins initiate those cells to undergo apoptosis. So that's actually how it works. It induces, radiation induces apoptosis in the damaged cells, damaged from the radiation. Now, if the cancer is there because the tumor suppressor system doesn't work properly, in other words, when it detects a cell that's damaged, it doesn't cause the cell to undergo apoptosis, then radiation therapy will not work for that kind of tumor. So there's one part of the tumor suppressor system is P53, protein 53. It's one part of the immune system. BRCA 1 and 2 are other parts of the tumor suppressor system. Usually when there's a mutation in the BRCA genes, women are at greater risk of breast cancer and ovarian cancer, and men are at greater risk of prostate cancer, among other things. So those are tumor suppressor genes, and that's heritable. And so if a woman or a man inherits BRCA mutation that inactivates that tumor suppressor system, they're more likely, they're going to be developing tumors all the time. That's what happens actually in our bodies all the time. We're developing very small, let's say, tumors or even malignancies, but our tumor suppressor system comes in and wipes them out. Well, if the tumor suppressor isn't working properly, doesn't wipe them out, then cancer radiation is not going to work in terms of killing the cancer. Now, then there's chemotherapy. You guys probably know people who underwent chemotherapy. Standard chemotherapy, what it does is it targets cancer cells that, let's say, might be growing faster and are more susceptible to this drug than normal cells that are not replicating all the time. So it generally works by killing, basically it kills the cancer cells faster than it kills normal cells. In other words, you're using a toxin that's more likely to work on the cancer than normal cells, but it destroys normal cells too, and that's why people who use standard chemotherapy, they often get very sick, hair loss, all that. Now, sometimes what they do is, remember, as cancer cells replicate and tumors grow, there's evolution in the cancer cells. They generate new mutations. It may be that a chemotherapeutic agent might work for a while, and then a few cells get mutations that make it less likely to work, just like an antibiotic resistance on bacteria. And so it stops working. So normally what they do nowadays is they often use a number of different chemotherapy drugs that target different parts of a cell cycle. And so if you have a mutation, let's say, that manages to avoid the effects of one of the drugs, it's unlikely you're going to have mutations for the other drugs that work on other parts of the cell cycle. So normally what they use now is a drug cocktail, a number of different agents. Just like people with AIDS, initially, the first drug they used was called AZT. It's an antiviral. It interrupts the viral replication process. And it worked for a while, and then those patients relapsed because there was a mutation where it didn't work anymore. And that's why now the HIV patients actually get a drug cocktail as well. And they use 3 different antivirals that target different aspects of the viral life cycle. And that's why a lot of HIV people who are positive are walking around today instead of dying like they used to in the past. They have very, very low viral levels because of the drugs that they're taking. But they still have the disease. If they stop taking the drugs, it would come back. Okay, so chemotherapy, similar. It's all about evolution again. In this case, it's cancer cells that keep replicating, keep generating new mutations. Some of those mutations are beneficial to the cancer cells. So, normal dividing cells are also often killed by standard chemotherapy, as I mentioned. It's because these are some of the cells that are continuing to undergo cell division. Like skin cells are being replaced all the time. So you take a standard chemotherapeutic agent that targets replicating cells, it's going to kill the cancer, but it's also going to kill other replicating cells that are just doing fine. And so that's why with standard chemotherapy, since you're using basically this toxic chemical, you end up with someone who gets really sick because of hair loss, nausea. Those are all because of being toxic to your cells, immunosuppression as well. Now, they've learned a lot more about cancer. It's a molecular disease, a molecular genetic disease, and I don't mean in terms of inheritance, but I mean in terms of mutations. And they've realized that they can target certain cancer cells directly without causing harm to other cells. And so this is called targeted chemotherapy. Good example is Herceptin, which targets an oncogene, HER2, that has a mutation in it. If there's that mutation, it's a very effective chemotherapeutic agent, and it doesn't cause all the problems. Unfortunately, I talked about this already, about tumor evolution. It may work initially, but it might not kill all the cells. Eventually, a mutation might arise that makes those cancer cells immune to Herceptin. Okay. Now, I talked about how cancer cells, like our own body cells, suppress our immune system. Our own body cells suppress our immune system so our immune system doesn't start attacking our own cells. It's a very kind of tightly controlled system. You want an immune system that attacks cancer and bacteria and all that kind of stuff, but not an immune system that attacks your own cells. So, because cancer cells are, like I said, they're a part of our own body. They're just cells that are replicating controllably now. They also suppress our immune system. And so, instead of our immune system normally knocking these cells out using apoptosis, those cells continue to replicate. Well, they now are they have a much better understanding of our tumor suppressor systems. And they're managing to get around this tumor suppressor thing that cancer cells do to protect themselves from the immune system. This is cancer immunotherapy. There's been some really dramatic results. For example, there's for melanoma, there's a checkpoint inhibitor. Remember a cell cycle? There's checkpoints at the G1 phase, at the end of G2, during mitosis. So it affects the checkpoint inhibitor system and allows our immune system to attack cancer cells. So that's what they use for melanoma. So before a person might, from the time that they've got stage 4 melanoma, from the time it's detected until the end, it's usually about 8 or 9 months. Well, that's been extended to like now 2 or 3 years, which is huge. In fact, some people, it's essentially cured them of melanoma. The trouble with checkpoint inhibitors is that, like chemotherapy agents, standard chemotherapy, it stops, it allows normal cells to be targeted by tumor suppressor genes, and it kills them too. So recently they've been discovering that a lot of patients who are getting checkpoint inhibitors, their cancer is getting cured, but their other cells are actually getting attacked by your own immune system, and that's actually killing them. One of the common ones is where it actually attacks your pancreatic cells that produce insulin, And so they're basically like type 1 diabetes. Patients who have their own immune system attacking their insulin-producing cells. Well, they've had that, so they've lost their insulin-producing cells, and they basically have type 1 diabetes, and they have to start using insulin all the time. So there is some downside to it. Now they're trying to get very specific with some of these new immunotherapies. There's other things like monoclonal antibodies that are specifically antibodies that are specifically designed to focus on cancer cells and those cancer cells that you have and not any others. There are vaccines that are similar to that. So normally what happens is with a vaccine for, let's say, a virus, vaccine is often made from either a dead virus, viral particles like bacteria and other cells. They have proteins on their surface, and normally our immune system, when we're being attacked by a virus, infected by a virus, there's these antigen proteins that our immune cells that recognize as foreign, and it mounts a whole immune response that kills our viral cells. And so when you get a vaccine for like a viral infection, before you get the virus, it ramps up your immune system so that if you then are exposed to the virus, your immune system is already charged up and go and kill the virus immediately. Well, you can do the same sort of thing in instigate a very strong immune response, but in this case to cancer cells, by making a vaccine for some of the certain proteins on the cancer cells that other cells won't have. And what's nice about this is, even if there's a mutation on these cancer cells and this protein isn't being made anymore and the vaccine doesn't work against it, you can actually make vaccines that are recognize multiple different kinds of antigens on these cancer cells. And so they just basically wipe out the cancer cells just like our immune system wipes out viruses. And this has been really successful. There have been some patients who've used this therapy. It's kind of in its beginning stages, where it's basically taken metastatic cancers from all over the body and shrunk them to the point where they're not there anymore. So things are looking up. Now, when standard chemotherapy or targeted chemotherapy or radiation or immunotherapy works, oftentimes what will happen is the cancer has stopped growing, maybe the tumors are shrinking, the patient's gone into remission. Sometimes it's temporary remission, it might last for a year or two, then the cancer comes back. So people are considered cured of cancer if they've gone 5 years without the cancer coming back. This is Patrick Swayze, an actor. He got, I think it was pancreatic cancer. Unfortunately for him, he went into remission, but then it came back and it eventually killed him. So sometimes there's even spontaneous remissions where someone has cancer, nothing's working, and then they suddenly go into spontaneous remission. No one knows why. So, different types of cancer. Now, there There are a number of reasons why certain cancers are deadlier than others. Some replicate more quickly than others, and they don't cause symptoms until it's too far gone. And so, let's say there's no early detection. So you might even have a slow-growing cancer, but you don't detect it until it's actually acquired enough mutations that it's now spreading around the body. Sometimes cancers don't lead to any symptoms that are detectable until it's too late, like pancreatic cancer, ovarian cancer is the same thing. Some cancers are easily detected, or more easily, like prostate cancers, because there's the PSA protein that's associated with damage to prostate cells. And you can measure that in the bloodstream. Also, a man who might have prostate cancer might have trouble urinating properly. And so he starts noticing that, so he goes to a doctor. Whereas ovarian cancer, usually it's not until there's some problem in the abdominal area, becomes uncomfortable, that it's actually detected. By that time, it's usually too late. One of the other things is if a cancer is not metastatic, that means it's lost contact inhibition, so it's invading surrounding tissue, but it's not traveling all over the place. Often that's a more treatable cancer than a metastatic cancer. The problem with ovarian cancer is it's a very metastatic cancer too. So not only does it grow in place, but it also generates tumors all over. No. Well, I actually mentioned earlier that there are some cancers that some organisms can actually catch. In other words, cancer cells can be transferred from one individual to the next. Dogs get this kind of low-grade cancer. It's not very aggressive. It causes these small skin tumors. It can be, in people, the dogs can get infected from other dogs. Tasmanian devils, a kind of carnivorous marsupial, there's a kind of cancer that causes these big facial tumors. They have very violent sort of sex, they transmit it to each other, and it is deadly. Clams also have kinds of cancer that is heritable. In other words, contagious. It's contagious. But for the most part, there's no cancers for humans at this point that's sort of been detected where you can catch it from someone else. Except, let's say, someone goes in and gets a transplant. And it turns out that transplanted organ has a small tumor in it, and then it's transplanted into a new patient. But that is a very, very, very rare occurrence. But, as I mentioned, there are viruses that are transmissible, right? That's pretty common. And one of those, HPV, human papillomavirus, causes genital warts, and for people who like oral sex, it causes throat and mouth cancer. But it usually doesn't happen, usually takes like 10, 20 years for it to happen. Before it was recognized as the causal agent for cervical cancers, cervical cancers are generally slow growing, but sometimes they're not detectable unless you go in for normal pap smears on a regular basis. And so that was responsible for a lot of cervical cancer deaths. But there weren't all that many, but still, especially for girls and young women, that was the leading cause. Well, there's now an HPV vaccine. It's usually teenagers, young teenagers, or prepubescent teenagers, boys and girls, might start getting it at between 10 and 13 years old. And it'll protect them from HPV infection. It's one of the most underutilized vaccines around because it's associated with sex. Now, we all know that having sex is immoral, that you're going to go to hell if you have sex. Even if you have oral sex. Same thing, you're going to go to hell. It's going to be terrible. It's really hot down there. It's better though, I think. I always like warm climates compared to really cold ones, and frankly, heaven seems just a bit too chilly for me. I also think there's more interesting people in hell. Usually better conversation. So if you're going to be stuck in a place for a long time, I think it's almost be better to be in hell and having great conversations than being in heaven where people say, "Oh, isn't God great? I love God. I love angels. I'm such a good person. You're such a good person. No one have sex. Sex is bad. Terrible." Okay. So, unfortunately, parents who get their kids vaccinated are obviously encouraging their children to be sinful, saying, "I'm giving you this because I don't want you to get the sexually transmitted disease," and obviously you can then go off and have as much sex as you want and not have to worry about it. And I think that's good. But some people are worried that their kids are going to go to hell for having sex, and so they're going to say, "Nope, no vaccine for you." And then the fact that you die from cervical cancer or something like that, or throat cancer, well, you're still going to go to heaven, right? Okay. So that's why HPV vaccine is underutilized. In some parts of the country more than others. It's, in fact, there's been, some real big things like, there was a governor of Texas, Governor Rick Perry. Now, he's evangelical, but even he was suggesting that all young girls and boys at the right age actually get the vaccine, but the Southern Baptists went haywire and, called for his impeachment. What can you do? Like I said, I'd rather have sex and go to hell than to heaven, but that's just me. That's probably too much information, isn't it? I know we reported now. Sorry, but this is actually a public health announcement that I'm giving. So perhaps you might even want to go in and get an HPV vaccine to protect you. Some cancers are heritable to a certain extent. In other words, there are germline cells. BRCA that causes breast and ovarian and some prostate cancers can be heritable. It's a tumor suppressor gene, so if it's not working properly and people start developing tumors, instead of the immune system wiping them out, it doesn't. There's another one. I believe it's sex-linked. Causes eye tumors. It's called retinoblastoma. There's two different kinds. First of all, remember, each chromosome is a homologous chromosome, one from mom, one from dad. It's a recessive genetic cancer. That means you need two bad copies for it to actually develop retinoblastoma. Okay, so this is a sporadic form where you need a number of different mutations over time, and usually it only happens in one eye, and that's the end of it. You lose that eye, but you don't have to worry about the other one. But the hereditary form is the one that's passed on. And let's say it's passed on to your offspring, and then there's a sporadic mutation in the same place, and now they're going to develop retinoblastoma. And retinoblastoma has something called penetrance. That's 90%. That means 9 out of 10 individuals who end up with it will develop retinoblastoma. The only cure, actually, is to take the eye out. And if you have the hereditary form, there's a good chance you're going to get it in the other eye as well, and you'll end up being blind. In fact, there's this guy named Daniel Tish. He's called Batman. When he was a little kid, he had retinoblastoma, lost both sides when he was by 18 months old. He somehow developed an ability, like bats, where he actually detect echoes coming back, and he could actually essentially see with his ears around him. And what he does now is he actually he makes these little clicks that he then receives back. He can ride bikes, he can walk all around, even in the dark, and it's just kind of like what bats do. And he's teaching it to a lot of young blind kids. It seems like you have to start fairly early for it to work. But anyway, so he had a retinoblastoma. Okay. And then BRCA again. So causes ovarian cancer, breast cancer, some prostate cancers. Angelina Jolie inherited the mutation, and she decided to have both of her breasts removed and then reconstructed. There's a debate about whether that women should actually do that or not. The chances of actually developing cancer with a mutation in the BRCA1 or 2 gene varies between 20 and 80% over a woman's lifetime. So there's a big debate about whether they should in fact have both breasts removed or just monitor. Okay.